Somewhere on a shelf in a hospital archive in Mongolia, in a drawer of glass slides nobody has opened in 40 years, there is a blood sample that should not exist. It belonged to a man who lived at the edge of the Gobi, in a settlement so remote that outside contact was rare even in living memory. When researchers finally sequenced it decades later, they found a genetic signature so old, so undisturbed by later migrations, that it matched almost exactly the blood chemistry of skeletons excavated from a cave more than 20,000 years earlier. A living man carrying, almost unchanged, the blood of people who died before the last ice age had even finished retreating. That is the strange power hiding inside something as ordinary as a blood type. It is not just a medical label. It is a fossil that happens to be alive, still circulating, still dividing, still being passed from parent to child in an unbroken chain that reaches back further than writing, further than agriculture, further than almost anything we call history. Most people glance at their blood type once, maybe on a donor card or a medical form, and never think about it again. O positive, a positive, B negative, 4 letters and a symbol filed away as a footnote of biology. But underneath that footnote is one of the most quietly revealing archives the human body keeps. Unlike a name, a nationality, or a language, a blood type is not something you choose, invent, or are taught. It is handed to you at the exact moment your existence begins, copied directly from the deep genetic library carried by everyone who came before. You, stretching back not centuries, but tens of thousands of years. And when scientists began reading that library seriously, using tools that did not exist even a generation ago, they discovered something almost nobody expected. The story of blood type is, to a remarkable degree, the story of Asia. Not Asia as a modern nation, not Asia as a line on a map, but Asia as the ancient staging ground from which entire branches of humanity fanned out to fill nearly every corner of the inhabitable world. To understand how strange and important this discovery is, you have to go back to the very beginning of blood typing itself, more than a century ago, long before anyone had the faintest idea that a red blood cell could hold a map of human migration. In 1900, an Austrian pathologist working in Vienna noticed something odd while mixing blood samples from different colleagues. Some combinations clumped into clots the moment they touched. Others stayed smooth, mixed freely, showed no reaction at all. It seemed almost trivial at first, a laboratory curiosity, but that scientist, Karl Landsteiner, had just uncovered the biological rulebook that would one day make safe blood transfusion possible, saving more lives across the 20th century than perhaps any other single medical discovery. He identified what we now call the ABO blood group system, and for decades afterward, the assumption among biologists was that type A, being so widespread across Europe, must represent the ancestral original human blood type, with H, O, and B branching off later as evolutionary afterthoughts. It felt intuitive, it felt tidy, and it was almost entirely backwards. And the correction came slowly, piece by piece, as genetics matured into a science capable of reading the actual instructions written into human DNA, rather than simply observing outward traits. Researchers eventually traced the ABO blood group to a single gene sitting on chromosome 9, a gene responsible for producing an enzyme that decides which sugar molecules get attached to the surface of red blood cells. Type A blood carries one specific sugar marker. Type B carries a different one. Type AB carries both. And type O, as it turns out, carries neither. Not because nothing was built, but because the instructions for building anything at all were switched off somewhere far back in prehistory, through a small mutation that quietly disabled the enzyme's function. In other words, type O is not a blank default. It is a deletion, a specific and traceable genetic event that happened to a specific ancestor at a specific moment in the deep past, and then was copied forward through every single descendant of that ancestor, all the way down to the people alive on Earth today. This detail matters more than it seems, because a deletion is something geneticists can actually date. Mutations accumulate at roughly predictable rates over generations, like a molecular clock ticking quietly inside every cell. By comparing the exact structure of the mutation responsible for type O blood across different populations on different continents, researchers realized they were not looking at one single event. They were looking at several occurring independently, or spreading outward from a small number of ancestral sources, each leaving a distinct genetic fingerprint depending on where and when it first appeared. And when the map of those fingerprints was finally laid out across the whole human species, one region stood out with startling clarity as the deepest, oldest reservoir of the mutation responsible for the world's most common blood type, the vast interior of ancient Asia. Consider what happened when explorers first began cataloging blood types among isolated indigenous populations of the Americas in the early 20th century, long before anyone understood the genetics behind what they were seeing. In many communities stretching from the Arctic down through Central and South America, researchers found something almost too uniform to be a coincidence. Type O was not simply common. In some populations, it was nearly universal, appearing in the overwhelming majority of individuals tested, with types A and B almost entirely absent. To a population geneticist, that kind of extreme uniformity is not random noise. It is the unmistakable signature of what is called a founder effect, the genetic echo left behind when a very small group of ancestors, carrying only a narrow slice of the full range of human variation, becomes the sole source of an entire population's later gene pool. Every descendant simply inherits what those few founders happened to be carrying, and nothing else, because there was no larger population around to mix new variation back in. The implications were staggering once fully understood. It meant that the ancestors of indigenous American peoples were, at some point, an extremely small band of individuals, and that band, whoever they were, likely carried a heavy concentration of the type O mutation before they ever set foot on the continent that would one day be called the Americas. And where did that small band come from? Every strand of evidence, skeletal L remains, tooth morphology, mitochondrial DNA passed down the maternal line, Y chromosome markers passed down the paternal line, and eventually whole genomes. But the story does not end at the edge of the Bering Strait. To really understand how deep the Asian roots of type O blood run, you have to look at what modern populations across the Asian continent look like genetically today, and then look even further back, into the DNA extracted from ancient human remains. Across East and Southeast Asia, type O consistently ranks among the most common blood types recorded, with some nations, particularly in the Philippines and across parts of Southeast Asia, showing type diversity found anywhere on Earth, shaped by tens of thousands of years of separate migrations, isolated mountain communities, river valley civilizations, and steppe empires that rose and fell for millennia. But underneath that surface diversity lies a deep and ancient reservoir of the type O mutation, one old enough and widespread enough to have seeded populations as far. A part is the icy tundra of Siberia and the tropical islands of the Pacific, thousands of years old. Techniques that once seemed like science fiction, capable of pulling a readable genome out of a fragment of tooth. The rise of the first agricultural villages and the birth of the earliest cities. And when researchers turned this technology toward blood type, the results were remarkable. Ancient individuals recovered from sites across East Asia, some dated to roughly third of Asia, long before the first river civilizations, long before the domestication of rice or wheat, long before anything we would recognize as recorded history. There is a second, quieter migration story tied to this same genetic thread, one that unfolded not across frozen land bridges, but across the open water of the Pacific Ocean. Roughly 5,000 years ago, seafaring communities believed to have originated near what is now Taiwan began a slow, extraordinary expansion into the Pacific, island hopping across thousands of kilometers of open ocean using navigation methods that still humble modern sailors, reading swells, star positions, cloud formations, and the flight paths of birds to find islands separated by vast stretches of empty water. Over the following centuries, their descendants reached the Philippines, then continued outward into what we now call Melanesia, Micronesia, and eventually the furthest reaches of Polynesia, arriving at islands like Hawaii, Tahiti, and ultimately Rapa Nui, also known as Easter Island, one of the most isolated inhabited places on the planet. When researchers examined the blood type distribution of Polynesian populations today, they found strikingly elevated frequencies of type O, echoing the pattern seen among indigenous peoples of the Americas on the no direct contact between them for most of human history, both carrying the unmistakable genetic fingerprint of the same deep Asian ancestry, arriving by two completely different routes, one across a frozen land bridge in the far north, the other across a chain of tropical islands in the far south. The ocean did not erase the connection. If anything, it preserved it, sealing each population off from later waves of genetic mixing that reshaped much of mainland Asia and Europe in the millennia that followed. None of this, of course, answers the deeper question that lingers underneath every migration story. Why did type O spread and persist so successfully in the first place? A genetic trait does not become the most common blood type on the planet by accident. It should be treated as on the Y chromosome. Because these two types of DNA are inherited without being reshuffled the way most of our genome is, they preserve an unusually clean, direct record of ancestry stretching back through unbroken chains of mothers and daughters, fathers and sons. When researchers examine East Asian populations, they consistently find D-deep, ancient branches of both mitochondrial and Y chromosome lineages, some tracing back 30 to 50,000 years, corresponding almost precisely to the same time depth at which the type O mutation appears to have become firmly There remain, even now, tantalizing genetic mysteries that researchers have not fully solved. Why, for instance, does type B blood reach some of its highest frequencies on Earth across northern India and parts of Central Asia, far higher than almost any else in the world? Some researchers have proposed that this pattern traces back to ancient population movements originating on the Central Asian steppe, a region that has served as a genetic crossroads for thousands of years, home to nomadic groups whose descendants eventually spread outward in every direction, carrying their genetic signatures with them into Europe, the Middle East, and South Asia. Others argue the elevated type B frequency may instead reflect a strong, localized survival advantage that Similarly, researchers have long puzzled over why type AB, the rarest of the 4 major blood types worldwide, appears somewhat more frequently across parts of East Asia than in Europe. One leading explanation is almost mathematical rather than biological. Because SS large parts of Asia and type AB can only occur when a child inherits an A marker from one parent and a B marker from the other, a higher baseline frequency of type B naturally increases the statistical likelihood D of AB offspring appearing across the wider population, without necessarily requiring any additional survival advantage at all. Some of the clearest windows into this ancient genetic landscape come not from bustling modern cities, but from small, isolated communities that have remained largely separate from the waves of migration and mixing that reshaped much of the rest of Asia over the last several thousand years. Certain indigenous groups in Taiwan, for remarkably close to what researchers estimate ancient Asian populations looked like tens of thousands of years ago, tea-like living time capsules, offering geneticists a rare and precious glimpse of what a much steppe migrations and the rise of vast mixing empires began reshaping the region's gene pool millennia ago. There's also a fascinating and still developing area of research connecting ancient trade routes to blood type distribution. Along the historic corridors of the Silk Road, the sprawling network of trade paths that once have been early travelers and traders along these routes, concentrations noticeably higher than what is typically found. Modern genetic science is still only scratching the surface of what blood type and ancient DNA together can reveal. New ancient genomes are being sequenced constantly, pulled from cave floors in Siberia, burial mounds on the Central Asian steppe, coastal shell middens in Southeast Asia and remote, island cemeteries scattered across the Pacific. Each new genome adds another data point, another small correction, another piece of nuance to a picture that is still very much being assembled. Some of today's leading theories about disease resistance, founder populations, and migration routes will likely be refined, revised, or in some cases, overturned entirely as the technology improves and the sample sizes grow. That is not a weakness of the science. It is exactly how good science is supposed to work, correcting itself, generation after generation, getting closer to the truth without ever claiming to have arrived at a final, unchangeable answer. What remains, underneath all of the ongoing research and all of the theories still being tested, is something almost impossible to fully put into words. Every person alive today carries, quite literally, inside every red blood cell circulating through their body right now, a fragment of a story that began tens of thousands of years ago on a continent whose deep genetic influence stretches from the frozen edges of Siberia to the sunlit shores of Polynesia, from the river valleys of ancient China to the remote highlands of the Andes. It is a story written not in ink, not in stone, but in sugar molecules attached, or deliberately, absent from the surface of a cell too small to see with the naked eye. And somehow, against every possible obstacle, that story has survived, copied faithfully from parent to child, across more generations than anyone can truly picture, all the way down to you, reading these words, carrying inside your own veins a quiet, unbroken thread connecting you to people who lived and died and endured long before anyone thought to write anything down at all.